All right, I think we got everything. I don't think we have any presentations in lab this week. So none of those. Just some housekeeping stuff we have. The IDEA evaluations are out. I didn't look to see if anyone's filled them out, but we're gonna do some extra credit on the course grade based on percentage that gets filled out. I'll get those posted. Our targets is always to get 90% plus. At 90%, we'll add 3 points to the overall course grade. That can help make a difference of a letter grade if you're right on that edge. But encourage you to fill it out. And as soon as you fill it out, you stop receiving the emails that say fill it out. We have exam on Friday. The exam will go through Plasmodium. It'll include the acanthocephalins and then all of the protozoans. So start reviewing those. The practical is on Monday. All right. So on that Monday, the lab will likely close 12 or 12:30. This oil immersion stuff could take me a while to find some organisms, but we cover the acanthocephalins and we cover the protozoans. All right. Life cycles, we really haven't had any life cycle quizzes posted. I'm now on the arduous task of uploading them in Blackboard, getting all the questions. Those should drop hopefully today, or by the end of the day tonight maybe. Those are not going to be for a grade. I'm going to have it set up as just entirely practice, so you can kind of take them over and over and over again to try to learn these life cycles. There's a lot of information on the life cycles, a lot of complexity to these life cycles. So those will drop. We do have lab this week, so we'll finish out. We'll look at toxoplasma and we'll see Plasmodium. Any questions? So we did our life cycles. Plasmodium life cycle, it is a rather interesting life cycle because the very first thing that happens is that those sporozoites invade the hepatocytes, the liver cells. They have to go into the liver cells and just have one round of replication. So they go through your merogeny to produce the merozoites, and then they get released, and then after that first initial stage, then they invade the red blood cells. All right, so we said that first part was a pre-erythrocytic cycle, or the primary exoerythrocytic zygony. Some of those that invade the hepatocytes will transition to become these hypnozoites, which are dormant stages, or the resting stages. Basically, sporozoites that just hang out. The cells, I guess, don't know that they're infected, but because they're there, they can be reactivated to start producing the symptoms of malaria again. All right? So after this first step of the hepatocytes, they then invade the red blood cells. We enter our erythrocytic cycle, and then from that point on, they will only infect the red blood cells. They'll go inside of them. They will feed on hemoglobin and whatnot. All right? Some of these merozoites that get inside the red blood cell will not replicate. They will just hang out in red blood cells, or if they replicate, they only replicate a little bit, and they don't really replicate very quickly. So that means then that there could be a time when all of a sudden these things switch and become your regular replicators, so then you start to get synchronous release in the red blood cells and a synchronous release of a bunch of merozoites that then produce the symptoms of malaria. We'll talk about that. So that's our pathology. When we have those reactivations, it's called recrudescence. All right, it's the initiation of clinical disease resulting from activation of schizons or merozoites in the red blood cells. All right, I also learned there apparently is a major discussion between schizons and merons. What's the difference between them? And they're really breaking it down to how they replicate. That's what distinguishes them. We'll just say that it's merozoites or schizons. We're just going to use them interchangeably until they resolve that confusion. But notice the difference. Relapse is reactivation of the hypnozoites. Recrudescence is basically reactivation, not necessarily reactivation, but all of a sudden uptick in replication in the red blood cells. And I've got these two species, Vivax and Ovale, exhibit this true relapse. Falciparum and Malari represents this recrudescence. All right, mosquitoes are the host, and once our female mosquitoes are infected, they are infected for life. And it's thought that infection will stimulate feeding, just make her bite even more and spread the parasite. All right, ready? We know a lot about apicomplexan and their entry into cells from Plasmodium. Now, this probably isn't universal across all apicomplexans, but we're going to utilize Plasmodium to talk about how these cells get infected. All right, so our invasion of the red blood cells is a very specific and sequential process. It happens regularly. It's very specific changes that have to take place in order for a parasite to get inside of it. So these are the steps. Number one is cell recognition. So you're going to have surface molecules on the merozoites. They're going to recognize the surface molecules on these red blood cells. If they recognize it as saying, hey, this cell is good, all right, they will bind and then they will orient themselves via a process called gliding motility. Gliding motility is actin-myosin driven. What it kind of does is initiates some of this movement to basically cause the merozoites, so cause the plasma membrane to dip a little bit and then bounce back up. That would allow it then to flip up in position where its apical complex is in contact with that plasma membrane. All right, so this gliding motility is actin and myosin driven. And that's the process of orientation on our blood cell. Once we get into position where the apical complex is in contact with the plasma membrane, we then have junction formation. So that's step number 3. So now we actually have binding between our apical complex, that part of our merozoite, and the red blood cell membrane. And then once we get this junction formation to where things can now be injected into the cell, then we have cellular entry. Now, the cellular entry happens in these 3 steps. First, the micronemes empty. So the micronemes are part of the apical complex. Those disappear, they empty. Next, the rhoptries empty. So micronemes, then rhoptries. And that's going to stimulate the invagination of our red blood cell, forming this vacuole and basically encapsulating our parasite inside of a parasitoperous vacuole. Once you have that, once you get into the vacuole, then the dense granules get released. All right, and again, we've got the micronemes, the rhoptries, these dense granules, those are all part of our apical complex. These dense granules, kind of unsure what their role is. We're already inside. What are they being used for? Not quite sure. However, this does induce some important pathological consequences, and we'll talk about them. Now, this is a very simplified view of how to get in. We know a lot more about proteins, pathways, what gets turned on, what gets turned on. We know a lot about that stuff. If you're interested, go through and take a look at it. But we're not going to worry about all these different pathways and so forth. So this is pulled from Parasitology Today, which was a review article. Parasitology Today is a journal. They had a review article on the entry into these red blood cells. So, pretty cool. Questions? Very sequential. Cell recognition has to know what type of red blood cell it's in. Then it orients itself via gliding motility so that our apical complex is in contact with the plasma membrane. Then we have our junction formation, and once we have the junction formation, this apical complex, everything in the apical complex starts to dump out, causing the plasma membrane to form parasitophorous vacuole inside of that red blood cell. All right. Plasmodium causes malaria. Malaria has a very characteristic pathology, symptoms. We call them paroxysms. So the paroxysms is the periodic symptoms and fevers that are exhibited by malaria. It's very characteristic. Right, so the periodic part means it's going to happen over and over again at a specific frequency. So let's talk about the paroxysm. So you're out, you're in a malaria endemic area. Heck, it might be the Everglades, who knows? You're in a malaria endemic area, you get bit by a mosquito. All right, you don't think anything of it. Then all of a sudden, you start to get intense feeling of cold that has convulsions and shaking associated with it. You have an intense feeling. That leads to a very high fever. Your body temperature just shoots up, 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 up. It ends up spiking at around 104 to 106 degrees, very high fever. All right. As you're approaching these high temperatures, you will start feeling nauseous. You'll probably be vomiting associated with them. And oftentimes delirium sets in. It's associated with some of these very high fevers. You're going to experience this for several hours, and then the fever is going to break. And when it breaks, you're going now to start to exhibit this profuse sweating because your body is going to be trying to drop its temperature, which it does, and it drops down to normal in about 2 to 3 hours. And then you're going to be on reprieve and be okay. But then this entire process again is going to take off anywhere from 48 to 72 hours later. Same progress. You'll go through this feeling of intense cold, convulsion, shaking. Your body temperature is going to start to increase. Fever is going to be spiking very high. You're going to start exhibiting the nausea, the vomiting, the delirium usually again, and then your fever breaks. This part represents the paroxysm, and it lasts 8 to 12 hours. So it goes 8 to 12 hours, and then you're basically going to have 32 hours, 36 to 40 hours break. So say 2-day, maybe a 3-day break before it starts back up again. And that entire thing is going to last 8 to 12 hours. Very, very characteristic. If you've known anyone who's had malaria, ask them about it. They'll have it. And we did. Several years back, we had a student from Africa. He's had it a couple times. And when we made it, he's like, that's exactly what happened. All right? So this graph kind of depicts what's happening. You've got your chills, peaks, then you've got your sweating. That's your recovery part. And then it's down, nothing. And this is your body temperature. So you're fine. All of a sudden your chills happen again, body temperature peaks, and then you go through your sweats and everything. And you can see that from start to start, it's going to be anywhere from 48 to 72 hours. It is very specific based on the parasite, which species of Plasmodium we have. All right, so on this graph, we have Plasmodium vivax. It has these very characteristic paroxysms. But falciparum doesn't. Falciparum, you get it, and you have almost no reprieve whatsoever. And the reason is that vivax has very synchronous release of these merozoites in the blood cells. The blood cells They almost all ruptured about the same time, which means you have a large number of merozoites, a large amount of cellular waste entering your body. You've got a very strong immune response that fights it, and then the immune response clears out what's there. Other merozoites enter red blood cells, and then it's a reprieve. Falciparum doesn't do that. Falciparum is much less synchronous. So you have the initial stages where the red blood cells rupture, releasing the merozoites and cell contents, but instead of all of them happening at once, you've got red blood cells that are continuously rupturing, which thus extends that period of time when you're having more and more of these antigens entering your body. Again, this is all the immune response. It's inflammation. It's responding to all of these merozoites and everything else that is being released. So in falciparum, falciparum is normally the deadly form, like the really deadly form of malaria, and it's basically because it's asynchronous. And there's partial reason for that, but there's less synchrony, no rest period, and so forth. And this part shows what's happening, where your merozoites are in the blood, they infect the blood, they're replicating. By the time they rupture, that marks the start of your chills. Any questions? This is malaria. All right, so with malaria, it's not just these paroxysms, but we see some other things that can happen. All right? We mentioned those dense granules. We're not quite sure what their role is. At least it doesn't seem like they have a role in entry of the red blood cells because they're already in. They're already in those parasitophorous vacuoles when they get dumped. But we do know that these dense granules induce a change in the red blood cell shape, the shape and the membrane structure. Again, and this is another one that you can look up, that we have a lot of information, a lot of pathways that are altered because of these dense granules. If you're interested in some of that stuff, you can look it up and read more about it. All right, so what are the changes? Number one, the membrane becomes a bit more porous, you can say. It's more permeable to small molecular weight molecules. And that's a good thing for the parasite. Parasites are in there replicating. Because they're replicating, they're acquiring energy. They need energy to replicate. So if you're making the membrane more permeable, you have more of these small molecular weight molecules like glucose that can diffuse right into that red blood cell and provide nutrients via glycolysis, because that's primary mode of ATP production for our parasites. So dense granules make sense. It's a benefit for the parasite. The other thing that happens is that you start to get these protrusions or these knobs forming on the membrane. All right, they're electron dense. That's how you see them in electron microscopy. All right, and these knobs seem to cause the red blood cells to stick to vessels, stick to the capillaries. All right, so it's thought that this allows the red blood cells to get stuck in the capillaries and the post-capillary venules, kind of keep them near the surfaces where they're more likely to be consumed by the parasite. I'm sorry, be consumed by the mosquito that needs to pick up the parasites. Is that what's happening? Probably not. Probably not, because you're going to it's not the merozoites that get picked up, it's the gametocytes that get picked up. All right, but they do. They tend to have some of this sequestration in the capillaries and post-capillary venules, and they do that because it changes the cytoadherence property, so it makes red blood cells stick to the vessel walls. Probably the thing that it does do is this. It hinders clearance of the red blood cells by the spleen. So the spleen's going to clear out damaged old red blood cells. All right. And that's not something the parasite wants if it's still replicating and still needs to finish getting into bloodstream so it can be picked up by the mosquito. So we know that these hinder clearance kind of keeps these red blood cells in the blood longer than needed. All of this stuff doesn't really do a whole lot for the host in terms of pathology, except in certain places. And there's a specific malaria called algid malaria, where it's about adhesion of these red blood cells in the capillaries that leads to blockage. So you could have pulmonary edema. So again, on the diagram, you can see an interferon gamma and tumor necrosis factor. Those are both pro-inflammatory cytokines. All right, these things get stuck. All of a sudden, they start increasing this. You can cause fluid buildup in the lungs. That's pulmonary edema, leading to hindrance of respiration. That would ultimately lead to excessive administration of IV fluids, which thus is a bad thing. And then algid malaria, which is basically the development of shock happening from all of this stuff, from the stickiness of these red blood cells. Normally, when you get the shock, it's going to lead to death, and it happens pretty quickly. And we do have vascular occlusion. We're going to talk about some of that stuff when it comes to the brain. Ready? All right, we have 4 species. 4 species differ in their pathologies, like how severe they are. All right, so we've got Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, and Plasmodium ovale. All right, I included this table, a blank table, in your notes. So the 2 most common forms of malaria are falciparum and vivax. Falciparum is cosmopolitan. It is most severe in the tropics and subtropics. Vivax is mostly temperate Plasmodium. It'll be in Asia, North Africa, and so forth. So it's not as cosmopolitan. These are the 2 most common malaria species in humans. But then we have 2 minor species. Plasmodium malariae infects about 7% or so, and then ovale is pretty rare. It does pop up as being in a human infection. Malaria is cosmopolitan. Ovale is basically in the tropics and most specifically on the west coast of Africa. So it's a much smaller area where you would find ovale. On that first step of red blood cell infection, you have to recognize the red blood cell. And we're going to talk about how they do that. But during that cell recognition, falciparum can recognize any type of red blood cell that's out there. I should say any age of red blood cell. They can infect young red blood cells. They can infect old red blood cells. And that's a great thing because you've got different age red blood cells cycling in your blood, right? And the age is not just like how old they are, but also cell receptors. The younger red blood cells have specific receptors. The older ones have specific receptors that the young ones do not, vice versa. Right, so falciparum can infect any age, which means you have a lot of red blood cells that they can infect. Vivax can only infect the immature red blood cells. So they have a much smaller pool, which, if you do that comparison, then Vivax will likely be less severe of an infection than falciparum, and that is 100% true. That is 100% true, and that's true based on their name. So malignant tertian versus benign tertian, or this tertian ague. So malignant, much more severe than the benign form. Malaria only infects the aging, which is an even smaller group of red blood cells than the immature red blood cells. And that probably is partially why you don't really see a large percentage of infections. And then ovale is unknown. It's a rare form. It's rare, so you don't really get a whole lot of money to study this one. With the paroxysms, we talked about how long it takes between activity, or between recurrence of it. So most of these are 48 hours. Falciparum, vivax, and ovale. So you've got 48 hours. At 48 hours, you start it, and then you're going to end it 8 to 12 hours later, and then you start back up that 36 to 40 hours later. Right, so every 48 hours, every 2 days, you start the symptoms again. Malaria is at 72 hours. Right, so 3 days. That leads to different names. So malignant tertian, benign tertian, ovale tertian, referring to the 48 days, which you'd say, well, hold on. Tertian kind of means 3 days, and quartan is the 4. Even though 72 hours is 3 days, where did that come into play? Well, it's just an old way of how they classified it. So it was like day 0, then day 1, and then day 2, and day 3, and so forth. But malignant tertian, benign tertian, all that, like malaria, what species causes malaria? I could say what species causes ovale tertian malaria? Plasmodium ovale. What species causes malignant tertian malaria? Falciparum. So they are specifics, and they use these specifically in medical terminology. And then our last bit is relapse and recrudescence. Again, one of them is from hypnozoites. True relapse, that's Vivax and ovale. The other one is from red blood cells. That's falciparum and malari. It's the red blood cells. So it used to be if you've ever had malaria, you are completely excluded from donating blood. Kind of comes down now to what species of malaria did you have, all right? Because if it's falciparum or malari, there's a limited time when they can come back. So normally I think it's excluded for 18 months. Because after you've had it and cleared it, if you went 18 months without having a reoccurrence, then you're considered clear. But if you've had Vivax or ovale, all right, those things are in your liver. And they could recur 30 years later, 30 years down the road. So on those, they normally exclude you completely from donating blood. So I say know the rows in bold. That's these 4. Make sure you know them. Ready? All right. Host defenses. There is some protective immunity that develops. All right. And that's our figure here. All right. That's part of our figure. So what we do know is that mothers in endemic areas are constantly getting hit by malaria. They have antibodies that they transfer to their infants, especially as they continue breastfeeding. They're transferring antibodies to their infants. Those maternal antibodies confer protection to these infants. So usually the first time these kids start to get malaria, it is after they finish breastfeeding. So after they've been weaned, since we're in biology. All right. So after they have those first rounds, if they survive those early rounds of infection and get through their early childhood, then we stay inside of those malaria endemic zones, what you typically see is premonition developing. All right, we talked about concomitant immunity. Now we're talking about premonition. We mentioned premonition before, but it's stimulated by small residual populations in the body that could lead to asymptomatic cases. So you have the parasite, they are replicating in the blood, but they're not replicating very quickly, and your body is not fighting it as aggressively as it typically does. So that leads to asymptomatic cases. What we also see is that those individuals that are in endemic areas are constantly exposed to the antigen, and their body constantly is primed for these parasites. So it keeps the infection from happening. It keeps the symptoms from occurring again. So children in those endemic areas will maintain their immunity as long as they remain in that endemic area. And that's why I said if they could survive the initial onslaught of malaria, then they're normally going to be good. They're normally not going to die of malaria. But If they leave the area for, let's say, a year and then come back, they have to go back through that process of building up immunity again. All right? And this happens. That was the student that was in the class. Went away, left the country, came back a year later, hit malaria again. It's happened. So premonition is important. It's demonstrating the protective immunity that we have for it. And that's kind of what what you typically see. So when you have premonition, your parasitemia is going to be pretty low in this case. When you have partial immunity, you're going to have high parasitemias, but how long that lasts, the parasitemia lasts, is determined by your body. So the reason you start getting this extended case of parasites inside the blood is your body, your immune system, stops responding as strongly to clear the parasite. And because it's not responding as strongly, your symptoms, your paroxysms will disappear. That's the asymptomatic portion. Why does the asymptomatic tolerance develop? It's probably to tumor necrosis factor insensitivity. So your body's constantly seeing this pro-inflammatory cytokine, it just decides, I'm going to stop responding. So even though tumor necrosis factor wants to be upregulated, your body says, heck with it. And thus you don't exhibit these paroxysms. It's great for transmission, right? Keeps the parasite in the blood for a longer period of time, more likely for the mosquitoes to pick it up. But that's a bad thing for humans because we're transmitting it to the mosquitoes. We're keeping it in the area. That's the first part of our host defenses, protective immunity. Any questions? What else do we have? Host genetics can play a role. It's been hypothesized that the Duffy blood groups evolved in response to malaria. What are the Duffy blood groups? It's your blood types, your A, your B, your O blood type. These types are characterized by specific antigens on those red blood cells. They are expressed on developing red blood cells. They're expressed on developing red blood cells. So, antigens A and B are recognized by Plasmodium Biology. Those are just on your immature blood cells. O blood types lack the antigen. This. They lack the antigen. So with this antigen, if you lack those antigens, you are pretty much protected against Plasmodium vivax. Because vivax only targets those immature red blood cells. They are targeting those antigens. And that's just kind of a schematic of what those are. It's kind of interesting, too, since we know that. 60% of African Americans have O-type blood. If we break it down and say, okay, let's narrow it down to those that were from Africa, have African descent, it's anywhere, it could be as high as 100% of those individuals. 88 to 100%, again, variation. But strong case for maintaining the selection pressure for O-type blood. It confers protection against vivax malaria. Doesn't confer protection against falciparum. So ready for that? So what developed in response to falciparum? Sickle cell. Sickle cell is mutation in the hemoglobin. There's multiple mutations that will generate this. We talk about the sickle cell gene as having two alleles to be normal. So AA, that's your normal red blood cell. When you're normal, you are susceptible to infection by malaria. If you have the sickle cell trait and you're homozygous, you are immune to infection. you cannot get falciparum malaria. Why? Because it alters the membrane and alters a pathway inside of it, which means the parasite cannot replicate inside those cells. So you're immune to it. The downside is that the individual usually dies before the age of 30. So how did it evolve? Oh, hold on. And so we have that. If you're a carrier, so this is a codominant trait. All right, it's incomplete dominance. If you are a carrier, some of your hemoglobin is normal, some of it is mutated. All right, so you have fully functional red blood cells, some of them. Some of them, they're mutated. They take that sickle cell shape. But those individuals are 80 to 95% protected against falciparum malaria. Again, the most deadly form of malaria that we have. So I think we can make a case that, again, selection pressures favored sickle cell gene. Those individuals that had sickle cell trait had protection. They were much less likely to get malaria. But in this case, these individuals died before the age of 30. How did it evolve at that point? How did it persist? How did it get how does it maintain? How is it maintained in our population? What do you think? More likely, yeah. Just play the odds. I'd also toss this out. Did this evolve like 100 years ago? Probably not. Our version of reality has changed a lot. All right. If you were 30, you're probably a village elder at that point. I mean, in human evolution, you just think about it, people rarely lived beyond into their 30s, 40s, and 50s. So in this case, you die before you're 30. It's probably not a bad, it's a bad thing. But evolutionarily, it can survive because a lot of the reproduction is happening in the teens, the mid to late teens and early 20s. So you still have a chance to reproduce, pass the traits on. Now, there's more pathology associated with the sickle cell disease. So normally you start to see there's a hindrance on endurance, hindrance on moving around, oxygen transport, and so forth. So that probably is arguing against this idea that, well, individuals are reproducing and they do that before they die. I think it's probably more of this case where you're a carrier, the chance that you actually have an offspring that is homozygous recessive is much lower than generating a carrier, a heterozygous individual, and that has allowed this trait to persist. There's a lot of research going on with sickle cell to try to, in some cases, cure it, right? Stem cells, stem cell transplants, and so forth, and also to treat it. So our prospect for life with sickle cell, lifespans have increased. A lot of cool stuff if you're interested in that. So, Duffy blood group, sickle cell anemia, and our last one is this thalassemia. Thalassemia is not sickle cell, but it is a defect in the hemoglobin formation that causes a decrease in the number of red blood cells. All right, and when the hemoglobin is malformed, the parasite can't really utilize it for metabolism. This is common in individuals from the Mediterranean region, the more temperate regions. This trait, this thalassemia, this disease, you have varying degrees of severity. So some of them are very severe. Infant might not survive beyond a few days of being born. Other cases, they'll live to early childhood. Some cases, you'll have it, you won't realize or you won't get a diagnosis until you're in your late teens or early 20s. All right, it's decreased red blood cells, malformed hemoglobin, and so forth. When you have this thalassemia, you show a decrease in susceptibility to all types of Plasmodium, 75% decrease. So this is probably another one of these diseases that developed and has persisted because of the protection provided against that Plasmodium. And again, Plasmodium's a killer. Plasmodium's a killer. It's one of the leading causes of death. Leading causes of death outside of the developed countries that have massive healthcare systems. So, host genetics, or the host defenses, this is what we've developed to fight Plasmodium. Any questions? Move on. All right, pathology. So there's other pathology to consider. We talked about the paroxysms, but there's other things that'll happen that are associated with it. Number one is that red blood cell destruction. You've had the replication in the red blood cells, they're going to lyse them, and that means all of that cellular contents are going to dump into the body. All right, so associated with malaria, with our paroxysms, would be severe anemia. All right, severe anemia. And it's not just the infected red blood cells that are lysed. Once some of these infected ones will lyse, you have inflammatory responses that develop and start to cause some of our uninfected red blood cells to be destroyed. The process of that is complement-induced autoimmune hemolysis. So it's complement-driven. Right. Why this happens? Hypothesis is possibly tumor necrosis factor toxicity. You've had a huge increase in this tumor necrosis factor that starts to drive destruction of our uninfected red blood cells. So if you're losing a bunch of red blood cells, your body can't keep up. Thus you exhibit severe anemia. These red blood cells, they are dumping the hemoglobin, and the metabolic process of these malaria, which they are consuming hemoglobin, leading to release of hemozoin. That hemozoin has iron in it. Our body cannot recover, which leads to iron deficiency. So you have to get that iron from your diet. You can't recover it from these ruptured red blood cells. So the anemia, somewhat, I put it separate because we hear about iron anemia. We've talked about that before. Iron deficiency anemia. Well, we have iron deficiency and we have anemia, but that severe anemia is really tied to the red blood cell destruction. And then the iron isn't because the parasite's consuming it. It's just we can't recover it. All right. So what happens to that hemozoin? Hemozoin gets picked up by macrophages. Once it does, those macrophages become useless. So normally with the macrophages, they can phagocytize things, process it, and keep phagocytizing other things. But once they pick up the hemozoin, that's it. They're done. That macrophage is shot. The only way to get rid of them is to deposit or get rid of the macrophage itself. So what that ends up doing is depositing, or it gets filtered out in organs and deposited in organs, leading to the appearance of grayish or blackish organs. So the hemozoin is on this dark color. It's not red. It's not bright red. All right. It takes on a grayish color. And this kind of gives you a comparison of normal versus a brain that has hemozoin deposited in it. Slightly grayer, slightly darker than what a normal brain should look like. So any organ could be affected. Could be the spleen, could be the liver, could be the brain, could be kidneys, and so forth. All right, and the other thing is black water fever. It's a common condition with falciparum. This is where our kidneys are filtering out hemoglobin and the other breakdown products. So it's filtering it out of the blood, it's depositing it into the bladder, which we then pass out as urine, and that's going to make the color of the urine appear like it's black or cola colored. So you've got, that's patient urine, they dilute it to get to that brownish color. That's a hemoglobin. Your body's doing that. It's also bad for the kidneys. It's very hard on the kidney because this usually leads to renal failure as you're depositing, as you're getting rid of the hemoglobin. Mortality rate when patients start to exhibit this symptom is anywhere from 20 to 50%. All right. We also have disruption of some of our other systems in our body. That's what I was checking. This was it. So you've got a slew of host inflammatory responses. That tumor necrosis factor could be driving that autolysis of the red blood cells. But we also know that red blood cell production, erythropoiesis, is also impaired. And it's thought that they also may be due to tumor necrosis factor toxicity. So not only are uninfected red blood cells being destroyed, but we're also hindering our body's ability to replace those red blood cells. So we're kind of, again, it's a buildup of these symptoms, buildup of these things that ultimately will cause the death in individuals. The other thing that we can see is this cerebral malaria. So the symptoms of this, you're exhibiting the paroxysms, but then all of a sudden you start getting this progressive headache. It could go to coma. Fevers could reach higher than 108. That's a major, major fever. You're going to exhibit psychosis, psychotic symptoms. Convulsion is typical in kids that exhibit this. Death could happen in as little as a few hours if left completely untreated. Now, the signs of this is often alcoholism. So acute alcoholism, like a lot of alcohol. You can do this, but chance that the kids exhibit this probably low unless they get into the liquor cabinet. But even then, those areas, you're not in endemic areas. The reason for cerebral malaria was thought to be due to red blood cell sequestration in those capillaries. So it's basically you're not getting oxygen to the brain, and you're basically then starving the brain of oxygen, leading to all of these symptoms and death ultimately. But we know that individuals that recover show zero mental deficiencies. So think about individuals that have gone through drownings or whatever, and there's limited oxygen that gets to the brain, or you have major accidents which cause swelling and oxygen deficiency to the brain. Normally you see some mental deficiency, neurological deficiencies that develop. We don't see that in cerebral malaria cases, which thus strongly argues against this idea that it's lack of oxygen. Instead, the prevailing view is that it's actually due to nitric oxide. So excess tumor necrosis causes this nitric oxide, which is one of our innate responses, innate immune responses. So nitric oxide gets released from vascular endothelium and vascular smooth muscles in the brain. It's that nitric oxide that produces these psychotic symptoms, the coma, the super high fevers, but it doesn't destroy any neurons. So if you clear from it, if you recover from it, your nitric oxide diffuses and is gone, and it leaves the neurons still intact. So that's kind of the idea for the cerebral malaria. When you exhibit the cerebral malaria, it is a very significant situation, like a major situation. So, and I do have this. DDT is good for me. DDT was commercialized 1900s, I think 1930, 1940 was when it was commercially available. We used it in the United States, clear out our malaria problem, or at least partially. We did Tennessee River Valley Authority, Army Corps of Engineers controlled flooding. We used it, and then as soon as we cleared malaria out of the U.S., then we said, well, no one else can use it. So.
I just got a question.
Yep.
I know that mosquito trucks still go around areas. What is used in them now?
They could be using a type of permethrin or an isomer of permethrin that'll target the insects. I don't know if they're mosquito specific or just generalist, but when I was working in college as a vector control technician, fancy term for mosquito control, yeah, I would mix up permethrin and fog it in areas, and it was always, it was non-specific. So any insect that had it, permethrin goes into the spiracles and then crystallizes and blocks their respiration. So it's possibly what they do. We also do targeted treatment at the larval stages at the water. So try to kill the larval stages. So they also do that as well. So. I would say, according to the, I ended the stream. According to the FDA,